Hello, everybody. We'll wait a few more minutes before we start our webinar today for with the Center for Analytics, and with the participation today and presentation by Dr. Lyle Muller and his lab at Western University. So bear with us maybe another minute or two as we allow a few folks to join and before we start. Thank you. Okay, let's get started. Welcome, everyone. This is our second edition of the Center for Analytics webinar series related to our analytics initiatives. And we're very excited today to have Dr. Lyle Muller and his team at Western University join us to present their work as part of the Center for Analytics and as an analytics initiative in developing really exciting algorithms and methods related to neural simulation and with potential impact on supporting the management of epileptic seizures. So we're excited to have them join us. Before we do so, I wanted to first begin with a few words and acknowledgements where OBI is located on this land, its nature and its people, and in particular the Anishinaabeg, the Mississaugas, the Wendat, the Haudenosaunee, Shawnee, and every other nation really who are recorded or unrecorded who have taken care of this land long before us. Today we have the important responsibility to take care together of this land, its plants, its creatures, and its people. And so before we start, I wanted to first provide a bit of background about OBI and the Center for Analytics. So I'll start with a bit of an intro on that, and then we'll hand it off to Lyle and team to really dive into the exciting work that they're doing with the Center for Analytics. So let me get started on a bit of that background. So OBI's focus is really to help accelerate and catalyze research in neuroscience on various fronts, developing biomarkers and understanding the molecular underpinnings of brain disorders is a key area of activity. Developing new treatments and technologies that support better diagnosing, better prediction, better potential treatment of brain conditions is also extremely important. Developing educational tools so we better understand these disorders, we can better train the scientific community, but also individuals who have brain conditions, and the community as a whole is also an important focus area. Finally, improving care. How do we translate the discoveries that are made? How do we translate the technologies that are developed so that we can have impact on positive impact on patients' lives? And so we do this through patient engagement, a lot of work around the informatics and what we call neuroinformatics as it relates to the brain. Commercialization, how do we support companies and startups in their early stages to really bring technologies to the bedside to support and have impact on patient health, and ultimately developing strategic partnerships. So those are all the major pillars and focus areas for OBI. And the way OBI does this is by implementing and integrating large research programs. We call them integrated discovery programs. And you see here on this diagram near the middle, these 5 large programs, including CanBind, that are looking at major depressive disorders, POND, the POND network, looking at neurodevelopmental disorders, the CP network, looking at cerebral palsy, the CALM network, a new program with OBI, looking at multiple mental health disorders in youth, and the Transcendent network, looking at concussion across the age groups. And each of these programs really integrate a number of partners from across the province of Ontario, but also Canada, and sometimes even outside of Canada. And this is what this diagram shows you, is all these institutions, and over 30 institutions collaborating and working on these large initiatives. And one of the big benefits that we've seen is in supporting these programs in collecting their data in a central repository, because this represents a really rich resource to better understand brain diseases. And so OBI created braincode now about 10 years ago, a little bit over 10 years ago, to support that integration of the data and enabling not only the primary purpose of these research groups to investigate and better understand brain disorders, but to also enable their collaboration with other groups and other groups to essentially access that data and develop their own research projects related to the data that have been collected. So we're really maximizing the value of that data. So the platform has over 1,000 users. We work on standardization, data federation, data linkages, and data analytics. So braincode overall has, had over about, like I said, about 10 years of experience so far, and so collecting data from these various programs over the years. So there's quite a few records from a number of individuals, over 28,000 records, over 13 million clinical data points. And it's really about multimodal data collection. We're collecting clinical data, imaging, molecular, genomics, wearables, and other data types that are part of these research studies. Data sharing is crucial. I mentioned this. We enable sharing within the research studies. There are often multiple sites involved, multiple hospitals and clinics that want to collaborate. Well, we do this through a centralized platform like braincode. We also enable that sharing with collaborators outside, which is really important. We also develop common data elements that enable that standardized collection of variables, not across all assessments or measures, but across a core set, what we call the common set of elements that span across these studies and enable us to then make more informed comparisons and analyses. Finally, data linking, and we use some fairly modern technology called homomorphic encryption to actually enable privacy-preserving linking of records, research records from these studies with other records such as health administrative data with partners such as the Institute for Clinical and Evaluative Sciences in Ontario, ICES. But there are challenges that still exist. It's been about 10 years that we've been working with, or a little over 10 years, as I said, working with the research groups and collecting this data, but we're still seeing a number of challenges related to the use of these data and maximizing the value of that data and developing the new techniques that we need to develop to really carve at their joints these brain disorders so that we can better understand them. So we need skill sets, first and foremost. There's limited expertise in advanced neuroanalytics and modeling, and the labs and companies, they really need that talent and the training opportunities. We need to accelerate how we develop these tools and enable access to modern technologies. So analytics and AI really is what we're seeing being developed and deployed even more and more in the research context, but also in the context of developing technologies that can help physicians and patients. And so we need to enable that compute access. Also the need for modern software that can make use of these more and more, the higher quantity of data and complexity of the data that are collected across these research studies and different sources. And we ultimately need to support the translation of these technologies so that there's a better pathway to commercialization, a better pathway to impact at the bedside for patients and the community. So we want to support these labs and startups and enabling also the compliance and the productization of these tools can be hard. So how do we do this? Well, we've created at OBI the Center for Analytics to address these issues. And our aim is really to help unlock the future of brain health via neuroanalytics, innovation, and translation. So that kind of sums it up. But in more detail, essentially what we're looking at is really developing those skill sets for the labs and the companies, enabling the better sharing of materials, enabling access to neuroanalytics and a community of practice that we can build where different groups can learn from each other and exchange methods, techniques, and knowledge about how do you address certain problems based on the types of data that you have and the type of either biological or health issue that you're trying to resolve. Acceleration is key, as I mentioned in my previous slide. That's a key challenge that we want to help address. And so we're providing access to advanced computing and high quality data on our platform with the Center for Analytics. And finally, translation is really important, where we want to provide the support to validate the techniques that are developed across projects, but also across groups that require the capacity to validate the new algorithm or new app that they might have developed that aims to support or help patients with their brain disorder. So regulatory and privacy requirements are also important, and ultimately advancing and supporting that advancement of these technologies into a productized state. So in a state where they can really be used in a real world setting. And when we think about the types of use cases that we see, we really think about areas such as clinical trials optimization, so CT optimization, where drug development often fails due to the heterogeneity of the data and the complexity of the conditions. So we need to be able to better understand the subtypes of the population so that we can design better trials and ultimately achieve better outcomes. Predicting these outcomes is also a challenge. So we want to develop better brain fingerprints to reveal patient trajectories. Comorbidity disambiguation is also a big challenge. The patients with brain disorders don't have just one condition, they often have multiple conditions. And we need to be able to disambiguate those using modern techniques. Treatment optimization is also a very important topic, and it's one that I think Lyle and the team will talk to us about today. But there is the potential for new techniques, new therapies that are non-invasive, using brain simulation and other technologies that allow for the better understanding of brain disorders and ultimately enabling us to develop new techniques for therapy. So thank you for that brief background on OBI and the Center for Analytics. We'll open up questions at the end of the session. But for the next little while, I want to pass it on to Lyle and the team to present on their work. And so I just want to introduce Lyle. Thank you for joining us today. Lyle's research really aims to develop new computational approaches to neural data and new mathematical approaches for neurodynamics. Lyle studied at Brown University. He completed a PhD in computational theoretical neuroscience in Paris with Alain Destex, and then did his postdoctoral research with Terry Sejnowski at the Salk Institute. Since 2019, Lyle has run a lab for computational and theoretical neuroscience at Western University, and now directs the lab for network science at the Fields Institute. So thank you, Lyle, and thank you, Mohammed and Lawrence, for joining us today.
Thank you so much for the introduction, Francis. I'll go ahead and share my screen. So you should be able to see my slide now.
That was great. Thanks.
Excellent. All right. So first of all, I just wanted to say thanks to Francis for putting this webinar together and for the opportunity to talk about our work today. So I'm Lyle, as mentioned, and I'm in the Department of Mathematics at Western University. And we've also just started this lab for network science at the Fields Institute. Before getting started, I just wanted to mention that we're happy to take questions throughout this session and to make it as much of a discussion as possible and as anyone wants. So please don't hesitate to unmute and ask questions while I'm talking. I'll give a brief introduction and motivation for our work, and then pass on to Lawrence, who will show our spiking network simulations and how to interface with that code. So just to get started today, I wanted to introduce the members of my lab who are here. So Lawrence Oprea is a postdoc in my lab who did a PhD in physiology at McGill. And Lawrence has been developing NetSim, which is our simulation code and the interface to that platform that we'll introduce today. And Mohammed Kesarwan is a neurosurgeon who's working on a PhD in computational neuroscience in my lab. Mohammed is working to combine his expertise in the clinic with computational analyses and models. And in the context of this project, he's helping us to figure out how these spiking neural network models can inform predictions on excitation inhibition balance and epilepsy. So I'll give an introduction to our work studying spiking networks, and Lawrence, again, will provide a quick introduction for how to run simulations using the code that I'll describe. So just to get started, I wanted to begin with the observations that got us interested in these large-scale spiking networks. And the specific observation was the dynamical activity pattern that we found in the visual cortex of the awake marmoset. So in this paper that was published in 2020, We studied LFP dynamics. That's the dynamics of the local field potential, which is the 1 to 200 hertz activity recorded on an electrode at the same time as the spiking activity. And we studied these dynamics that were recorded on these Utah arrays that were implanted into area MT of the awake behaving marmoset. And so by carefully analyzing the activity patterns with new signal processing techniques that we developed, we demonstrated that the ongoing activity, just the spontaneous activity in this visual area, is very often organized into traveling waves of neural activity. This here shows time in milliseconds in the moments just before the onset of a stimulus. And the local field potential here is plotted in color, with pink colors for negative potentials and blue colors for positive potentials. So in this work, we demonstrated that these waves of activity affect neural excitability, so the excitability of local networks as these waves are passing over the area. And we could also show that they affect perceptual sensitivity. In addition, we showed that similar waves also occur during normal viewing of natural scenes. But this demonstrates why these are interesting and important phenomena. But we also wanted to know what could be the network mechanism underlying these activity patterns. So with this in question in mind, the first thing we became interested in was their speed. And we observed here that the distribution of phase speeds falls consistently between 0.1 and 0.6 meters per second. These are for 2 subjects in the study. And this range was really interesting because this range of speeds falls right within the range of conduction speeds for the long range, unmyelinated horizontal fibers that connect neurons across single regions of cortex. So if you imagine that this schematic here is one region of cortex, there are fibers that connect neurons from one patch here up to 6 to 8 millimeters across that cortical area. Because they're unmyelinated, the conduction of the spikes down those fibers is slow. And we hypothesized that these horizontal fibers might provide the underlying network mechanism for these waves. And so to test this hypothesis, we started studying these large-scale spiking networks with excitatory and inhibitory leaky integrate and fire neurons. They're very simple neuron models that allow us to simulate large-scale networks. These neurons are operating in the balanced regime, and they're arranged on a 2-dimensional sheet here, connected with local random connections. So we made connections in the network distance dependent, with connection probability falling off as a Gaussian with distance. And importantly, the axonal time delay between 2 neurons grows linearly with the distance between them so that we could model the transmission of the spike. The other neurons in the network are also connected following these 2 rules. And importantly, we were studying the network at the biologically realistic scale, which means we needed to study these networks as they scaled from 10,000 to 1 million units, always keeping the same neuronal density at each scale. And these networks then have connectivities ranging from 1,000 to 10,000 synapses per cell, which is consistent with the dense connectivity in cortex. Reaching this scale of 1 million neurons with 10,000 synapses per neuron is really important because it means that the individual synapses can be weaker than in smaller scale networks with reduced connectivity. So this is a highly technical detail, but it's important because simulating smaller networks with stronger synapses, as is often done, can lead to high correlations that don't match the activity dynamics of networks in vivo. What I'll show later on is that by matching the correct dynamical state of the network, we will be able to start to make quantitative and model-driven predictions about the effects of both sensory stimuli and clinically relevant electrical stimuli. So achieving the simulations at this scale was made possible by a set of specialized C codes that we called NetSim, which is approximately just 1,000 lines of code, but that enables efficient and scalable simulations of these spiking networks. So time delays, again, coming back to this point here that we included time delays between the neurons here. And those aren't often considered when modeling neural networks. But in our work, we found this microscopic property has a profound effect on shaping the dynamics into traveling waves of spiking activity that are propagating over the network. So this depiction here is what I'll call a network raster plot. Here, there's time on the horizontal axis, and then cells across a one-dimensional ring along which all the neurons are arranged here. This type of plot is useful for visualizing how the spiking dynamics in the network evolve across the network over time. So plotted here are spikes over 800 milliseconds from 80,000 excitatory neurons. We haven't plotted the inhibitory neurons here. And the spiking here is driven only by internally recurrently generated activity. So we find that time delays shape this activity into these lines in the space-time plot that you can see here. And those represent these waves moving across the system, just similar to what you saw in the experimental data. Now, importantly, even though the dots may appear large here, only very few neurons are actually spiking as the wave goes past. And again, through this large-scale simulation code, we're able to find that networks of 100,000 to 1,000,000 spiking neurons emit a new activity state, which we called sparse waves. And the key detail here is that, again, very few neurons are spiking as the wave goes past. For example, raising the background spike rate in the network, if this is 5 spikes per cell, it will raise the background spike rate in a local network from 5 hertz up to 8 hertz, and then maybe back down to 2. So it's a weak modulation of the background activity or the ongoing activity in the network, which shows up as these kind of faint, more dense patches of spiking activity plotted here. We can also simulate the model in 2 spatial dimensions. And then using a biophysical proxy, we can visualize what these traveling waves of sparse spiking activity would look like in the simulated network LFP. I'll play this one more time. And you can see here this relatively negative potential propagating across the network there. So these results provide a clear example of how we can connect analyses of neural data. Here, for example, the LFPs that are recorded in an experiment, all the way to the mechanisms of neuronal spiking and synaptic input through these large-scale spiking models. And the model here also provides 2 specific predictions that we can then test directly in the Utah array recordings. So first, locations with relatively negative LFP will correspond to more spontaneous spiking. So the locations, as here, will have relatively more spontaneous spiking. For convenience, we'll call these depolarized states in what follows. Also, locations with relatively positive LFP will have less spontaneous spiking, and we'll call those hyperpolarized states. So this first prediction comes from the fact that the waves traveling through the network create changes in the balance of excitation and inhibition at different phases as they're moving across. And to explain in more detail, if we look over half a second of one simulation here, and during a time when waves are occurring in the network, we can see that the firing rate of the excitatory neurons in a local population is fluctuating in counterphase to the simulated LFP. So as the LFP becomes more positive, the excitatory firing rate is going down, and vice versa. And this means that in the network model, relatively negative LFP potentials correspond to a depolarized phase, and positive potentials correspond to what we'll call a hyperpolarized phase. So now to test this first prediction from the model, when we quantify spontaneous spikes as the waves propagate over the Utah array, we find that ongoing multi-unit spiking during detected traveling waves is indeed higher around plus or minus pi radians. That is at the negative potential, or again, what we call this depolarized state. And it's lower at 0 radians, which is the positive potential, or what we call the hyperpolarized state. So this observation verifies the first prediction from the model. And leading from this, the model also makes a second prediction, that these depolarized states will exhibit higher responses to incoming inputs. And we can illustrate this by stimulating the network during each state and then measuring the response. So this plot represents a set of simulations of the network model with precisely the same initial conditions and connectivity. In a case where there's no stimulation, which is this black line here, with stimulation during a depolarized state here. For example, if we sent spikes to a point of the network there, and with stimulation during a hyperpolarized state. For example, if we send spikes to a point in the network there. These are just random Poisson spikes over many trials that are plotted here, with the trial average plotted in this solid line here. So I won't go into detail here, but we're able to quantify the responses in the experimental data in a similar way. We were able also to confirm this prediction in the data. Specifically, we were able to show that depending on the alignment of the waves with the incoming spikes driven by a visual stimulus, the evoked response could be higher or lower in each state. And because the network is in the correct dynamical state that we think matches what the neurons at each point in the actual cortical networks are feeling in terms of their inputs, the prediction from our model matches what we see in the data to the order of magnitude. So specifically, if stimulus evoked activity reaches about 30 hertz in the depolarized state in this model, the activity in the same stimulus reaches only about 20 hertz in the hyperpolarized state. And that difference between the two states matches what we saw in the experimental data. So this now allows us to ask whether we can create predictions for other types of stimuli, including the types of electrical stimulation that are important for the clinic. So specifically, in this new project that we're working on together, we wanted to think about modeling one area of cortex in the non-human primate using one of these topographic balanced spiking network models. And we focused on modeling the response of these biologically realistic network dynamics to transcranial electrical stimulation, or TES. So here we simulated network activity just as before. You can see here the spikes in the network, plotted again as these black dots here. And we're also going to look at the overall firing rate in the network, which is plotted in blue here. We then applied a simulated TES stimulus. And we're going to study how the network's firing rate changes over time in response to the stimulation. You can see it here as the stimulus turns on, the network creates these dense bands of spiking activity, representing the network firing rate going from a baseline of 3 hertz up to about 12 at its peak. To do this, we used a biophysical proxy developed by Yuri Buzaki's group at NYU. And this proxy provided a way to translate the electric field strength from the stimulus into a current injection into the cells of the network. And the reason why this is important is that transcranial electrical stimulation often is a very weak effect on the neural activity. For that reason, it's often been dismissed as a useful and viable clinical opportunity for non-invasive stimulation. We believe that by stimulating the networks at the correct time and guiding the stimulation in a closed loop fashion, that we may be able to identify times, just like with the visual stimulus, where the network is more receptive to this electrical stimulation. And if that's the case, we may be able to use the relatively weak non-invasive stimulation of TES and may be able to guide our experimental colleagues in order to say, okay, if you stimulate here, we think that you'll get a stronger effect. So I think that's an important point because it shows how through the spiking network models and understanding the activity states of these networks, we can actually leverage non-invasive stimulation in interesting and new ways. Just to turn to our recordings from our collaborator, Chris Pack at McGill, this plot here shows multi-unit spiking activity recorded on an electrode across many trials. Here, this is about 300 trials recorded on one day. And this is, again, multi-unit spiking activity recorded from a single electrode. We're going to analyze the spiking activity and see how it relates to the LFP modulations I showed previously. So what we'll do here is to have a chance to make direct comparisons between the LFP modulations generated by the spiking network and use this to make model-driven inferences about the state of the network and how it will respond to externally driven electrical stimuli. So we can start with the LFP recorded in one trial, and then the spikes recorded on this electrode plotted here in gray. We can then quantify the LFP activity, as before, to understand how spiking activity is varying with the network state. So this is a plot similar to what I showed previously, showing that, as before, we observe more spiking activity in the depolarized state and less spiking activity in the hyperpolarized state. Importantly, these modulations we observe here are similar in magnitude to those we observed in the previous work. So it's the same cortical area and similar recording technique and a different species. This is in the marmoset and this is in the macaque. But this now puts us in a strong position to make model-driven predictions for how TES stimulation will affect spiking activity in the brain networks in vivo. Because we're starting from a solid understanding about the network's spontaneous activity state in order to motivate predictions about how TES stimuli can influence the system. So in conclusion, large-scale spiking neural networks produce activity consistent with that observed in vivo. These network models can provide specific mechanisms for neural dynamics observed in experiments. These network models can also generate experimentally testable predictions for responses to sensory and electrical stimulation. And finally, NetSim provides a fast, simple, and efficient interface to simulate spiking neural networks on standard workstation hardware. So I'll take any questions that you have now before passing over to Lawrence, who will show you about the software interface that we have to our NetSim code.
I don't see any questions yet. If anybody wants to unmute, feel free to unmute. In the meantime, I'll have a quick question for you related to how do you see this the approach of using TES having a potential benefit impact versus the current gold standard for treatment?
That's a great question. So there's a variety of new techniques that have been developed, and there are possibilities, for example, with vagal nerve stimulation or other possibilities for disrupting epileptic seizures at the onset. I think that TS is a really promising opportunity for stimulation because you can have a non-invasive stimulation directed directly at a specific brain network. And this technology is being rapidly developed. For example, there was a work just recently showing in a modification of this technology, strong activation and so strong modulation of the activity in human hippocampus. So it's a really promising technology for modulation of even neural activity in deep brain structures.
I can add from a clinical perspective, developing non-invasive methods is what a lot of clinical groups are going towards. Surgery is expensive, it's limited, it has its risks. So developing and really evolving these analytical toolkit that clinicians could have to use non-invasive methods is what I believe the gold standard of all technologies in medicine are going towards. We went from historically, surgery has been going on for thousands of years. In the 90s, we really optimized surgeries that continued. Now we're moving minimally invasive, and really key transition is non-invasive, and evolving our analytical techniques in evolving these methods is, I think, key in clinic.
Thank you. That's a great point.
Great. We have a couple questions quick online, and then we should give time for Lawrence to jump in. One from John Griffiths. Does the spiking LFP anti-correlation imply that the LFP oscillations are anti-correlated with metabolic load?
That's a great question. I'll mention that the spiking LFP fluctuations that we're looking at here are on a fairly fast time scale. So we're really on the tens of milliseconds time scale. And that's where we see these changes in these large changes in the excitability of the networks and visual cortex. There may be areas in, for example, frontal areas that have fluctuations of spiking activity that are on longer time scales. But because these are really fast, I'm not sure that what the specific link between the LFP fluctuations and metabolic load would be. But it is something that would be interesting to study in future work. There may be possibilities for looking at this with neuroimaging.
That's great. And one from James Ellis. Do you think human iPSC cell derived neuron networks might behave similarly to the ones that you've observed so far?
That's a great question. And we've actually looked at this a little bit with Dr. Ellis and others actually to try to understand this. So one of the things that's really interesting is that we have these activity states in the networks. For example, here, and this looks like just a sea of spiking activity. Again, every neuron spike is plotted as a little black dot here. What you're seeing here is this low rate, irregular, and low correlation firing state. The reason that that's really important or interesting for the spiking neural networks is that through work that was done through the 90s and early aughts, people started to figure out that this state was the one that matches what people recorded from in vivo. So if an electrode is implanted into a awake behaving subject, this is the activity state that we expect is actually happening in that 100,000 neurons around the electrode. This is actually produced through a dynamic interplay between excitation and inhibition. So instead of neurons collecting a bunch of spike or inputs together and then becoming more and more depolarized until they get to their spike, we believe that neurons in vivo are actually in a much different regime. They have very strong excitation and then very strong inhibition. And those are pushing against each other and approximately canceling out all the time. When the neurons are in that state, you get this type of activity regime where at the network level, you have this kind of sparse and decorrelated activity. I think it's really interesting for the possibility of human iPSC networks to start to recreate these activity states. Because if we can make strong links between the activity states in iPSC networks that look like this and what we see actually in vivo, ideally through spiking neural network models, I think we can actually help to translate the activity patterns that are seen in iPSC networks, how they change between wild type networks and those cultured from patients with disease, for example, and understand the links between how the spiking activity is changing at the network level. So that's a long-winded answer to say that I think that the human iPSC networks can behave similarly, but that we need to pay attention to really careful balances between the excitation and inhibition in these networks in order to achieve activity states that are matching those in vivo. But that's a great question.
That's great. Thanks, Lyle. So we'll maybe pass it on to Lawrence, I think, at this point.
Sure. We'll stop
Could I
my
ask
share.
quickly about how much time I have left?
So we have 20 minutes total, but it'd be great to leave I think 5-10 minutes for further questions. So maybe aim for 10 if possible.
Okay, should be
Great.
good.
Thanks.
Can everyone see my screen?
Yeah, great.
Okay, so I'll be doing a quick run through of the features of NetSim. But of course, if you have any particular questions, let us know here or in emails afterward. I'll be featuring two versions of NetSim. One is the public-facing one that's available now for anyone to use, and also we have a feature-rich development version of NetSim that I'll be going through afterward. So NetSim can be found here at our lab's GitHub page. Fastest way to grab it is either downloading the zip file, or you could git clone through the command line to get it onto your system. Once you have NetSim downloaded and unzipped, it is written in C, so it has to be compiled first. You would just have to run these two commands here, the two make commands, in order to compile it before it could be used. Now, the simplest way to run NetSim is through the command line interface, which there's an example here. You give it a parameter file, which tells it how to configure the network and how to run it, and where it should output to. Very straightforward. So I will begin a run here in my command line at the bottom. There we go. So initializing, etc., etc. This should take about 2 minutes. So I have time to go over the parameter files, which is determining the activity of this particular test. So here we have an example of a parameter file. It is structured extremely simply. You just write the name of the parameter on the left, and then an equal sign, and then the value of that parameter. So I have a PDF document here, which shows all of the possible parameters. Compile arguments on top, we won't talk about that right now. But here are the actual parameters. So for example, you have neuronal parameters like time constants, threshold potential capacitance, etc. All the units are in SI-based units for the sake of simplicity. You have synaptic parameters such as delay, which is, of course, is very important, conduction speed, excitatory inhibitory conductance. Network parameters, we featured N, the total number of neurons, and the number of connections, K in the talk as well. Some others here. And also simulation parameters. So for example, the total amount of simulation time, the time step for simulation, etc., etc. One thing I do want to focus on briefly is this connector parameter, which describes the topology of the network that you wish to simulate. So this is quite extensible, but at the moment, we have options for 1D randomly connected networks, 1D Gaussian connected networks, two-dimensional networks with Gaussian connectivity, or more exotic kinds of connectivity. For example, we have a project that features clustered connectivity in both the excitatory and inhibitory populations, which interact with each other to produce specific dynamics. And there is also the option for multiple cortical layers, if you want, for modeling either within a column or between different areas. So that is a simple selector there. Let's go check back in to the simulation. So it has finished. One thing is we found that the total number of spikes created during the simulation is a good signature for the simulation because the exact spike count is extremely sensitive to the parameters and the initial conditions that you have, but completely replicatable for any given random seed. So for example, this one gave 343,000 spikes. And if we look at the parameter file that produced it, you can see the expected output is 343 as expected. So this is the way that you can have tests and replicability. Okay, so what this actually did is it produced a series, which I hope you can see big enough here, a series of output files, which include things like voltage, conductance traces, and the spike counts, the individual spikes of particular neurons and the IDs of those. So as an example, if you would like to analyze the output of this in MATLAB, you can open up MATLAB, and we have pre-written functions for loading these data. So let me load the one that I just ran, and then I can make a scatter plot. So this is a scatter plot very similar to the presentation you just saw with neuron IDs on the y-axis and time on the x-axis. Okay, so that is the basic interface, outputs, and analysis pipeline. I want to feature the development branch in the time I have left because we have a lot more features there, and this is soon going to be updated to the main NetSim branch that everyone can use. So the largest change, except for significant performance upgrades in the development branch, is PyNetSim, which is a Python wrapper for NetSim, which should be comfortable for anybody that has any experience with modern Python toolboxes and the like. So the NetSim object, that the NetSim network is thought of as an object that has methods like running, analysis, etc., which should be pretty straightforward to understand. You can install it using pip. It's a package, and then you import it, and you define a simulation object, which is a NetSim network that is given a parameter file, directory, where NetSim is, output, etc., etc. So I will do this live. So this is creating a NetSim network with a particular parameter file at this location, which is given here. So we should expect about 34,000 spikes in this network that I'm testing. This interface allows for changing parameters easily through the Python interface. So for example, I'm modifying these two here. You can write out the parameters in the parameter file, or you can just add them here or modify them as you wish. And then the network has a run method, which produces the same kind of text output, and it's already complete here. So as expected, 34,000 spikes in this test network. We have also added a series of analysis functions. So for example, I am looking to analyze this network that I just produced by looking at the pairwise spike correlation, by looking at the coefficients of variation, and by looking at the firing rate. And you can just list them. You can list however many you like. It's a bunch of common analyses where you can add your own and then import them here. So I'll just do that quickly. So all three of those will run, and then the results are stored if you want to analyze it in Python, or if you want to export it and analyze it perhaps in MATLAB afterward. And this is just a list of different options for analysis. Coefficient of variation, binning the spike train, firing rate, making raster plots, correlation, power spectrum analysis, etc. So common things like that. There is also an option to do a suite of analyses. For example, Dr. Muller mentioned the importance of the asynchronous irregular regime in networks. There is an option in PyNetSim. You can just type in AI for asynchronous irregular, and it runs through a series of these different analyses on your output, and then you can judge based on those outputs whether or not you're in the regime, the asynchronous irregular regime. I also just have some quick plots to show what these analyses did. spike correlation, the coefficient of variation, and the firing rate, just as an example of how you could use this in a pipeline. Very briefly, I'll talk about the last major feature we have, which is called NeverSim. So this is a implementation of the NeverGrad, hiding it up top here. This is an implementation of the NeverGrad gradient-free optimization platform developed by Facebook. The fact that it's a gradient-free optimization allows you, it's convenient for these kinds of expensive to simulate networks. It might take several seconds or minutes to simulate a network. So these are the ideal optimization tools for that. And the interface is very straightforward. You define the parameters you wish to look at. You define a cost function, which can be based on these analyses you have here. For example, perhaps you have a target firing rate that you want, let's say 5 Hertz or so. You can set that as a target, and then NeverSim will automatically use NeverGrad to optimize your network to give you the parameter set that is as close as possible to that 5 Hertz target. And NeverGrad has a gigantic list of optimizers you can use. I'm just using 1+1 here as an example, but the documentation is quite thorough on their website. And it's all parallelized, it's all optimized and scalable to the amount of compute power that you have. I think I'm out of time, but I think that pretty much covers
that's fantastic. Thanks, Lawrence. So yeah, we'll open it up for a few minutes for any questions for Lawrence and about the tool and the simulation.
I'll just mention as well that there's a manual on the available. So first a quick README that goes through exactly the introductory steps that Lawrence mentioned in the first part. And there's also a more detailed manual as well.
Hi, I had one question.
There you go. Mark is unmuting. Great.
Great. Thanks. You provided some options for topology that were fairly standard, Gaussian, various kinds of Gaussian and other things. I'm very interested in the very heavily skewed distribution of network connections, and also in the prevalence of recurrent motifs. It's documented by several labs where it's more probable that you get a back connection, or more probable that you get a more than expected number of connections in 3 or 4 neurons, and that they may have higher synaptic values than you'd expect drawing from the marginal distribution. Is there any way to ask for those kinds of things in your simulation?
So the network topologies available so far have been developed on kind of an as-needed basis for the projects that we've been involved in. Any other kind of topology would be easy to implement, because all it does is kind of loop over all the neurons and assign some kind of target to them. And the way that target is assigned is completely open. It is at the C code level for the sake of efficiency. All the heavy processing is at the C code level, so it would take someone with C knowledge to implement it, but any particular topology should be implementable.
Okay, perhaps I'll contact you privately about that.
Sure thing.
Thanks.
Sure. And so I can even show here a quick example of, so this is just a quick example, just showing, so all of the—
Could you expand that just a bit?
Sure. Let's see.
Thanks.
That might make it a little bit more legible. Okay, so all of the connectors in the network, so this is just the random connect function. This is an example, but all of the connection functions in this software are in one section of the code. And for example, this is about 30, 40 lines of C. And so this is what you could write to implement that, for example, either the motifs or the log normal distributions. And so we think about this as something that's very extensible. The core code is about 1,000 lines of C. It's because we've written the interface, we've written it in a way that it's fairly templatable, and you should be able to ideally go in and modify those small sections where if you're interested in a log normal distribution, that you can plug in that code. And that's exactly where we're trying to go in the future development, is to make it more and more extensible along the lines of exactly that.
Okay, thank you. I'll send more details by email. Thanks both.
Thank you. Yeah.
I don't see any immediate other questions, but if anybody else would like to unmute, feel free. I had a quick question, Lyle. Are you and Lawrence, are you guys sort of enabling the community to contribute code to the platform at this stage, or thinking about doing that in the future, or if
That's a great question. We've
yeah,
been
Mark might create a new topology or a new algorithm for the topology. Could he contribute to that?
sure. Yeah, and so the code is available on GitHub and allows people to download and contribute. So that's definitely possible. We've been managing kind of the updates and everything, but if anyone's interested in contributing code to it, we can certainly integrate that into the main development releases. And yeah, we've been working to develop a kind of integrated feature and testing paradigm. And yeah, we would certainly welcome that by anyone.
That's great.
One last thing I'll mention as well is that so these spiking neural networks are often simulated on, large scale high performance compute architectures. And, even 5 or so years ago, it was thought that these types of network scales, millions of neurons that are applicable to the activity states in vivo, it was thought that you really needed to have a supercomputer or other large scale equipment in order to simulate them. And what we're finding is that through the specific implementations that we've done in C and the specific ways that we've structured the flow between the memory and the CPU in these systems, we can simulate very large scale networks on standard workstation hardware. So what Lawrence showed today just is the demo for the NetSim was on his laptop. And on the fairly standard fairly standard hardware that's provided to us by the OBI and Center for Analytics, we're able to go to these types of large scale networks of millions of neurons. And I think that in combination with the optimization techniques that Lawrence showed as well at the end of his talk, these never-grad optimizers, we can start to work with spiking neural networks in a way that's totally different from the way that I started when I started doing my research work in this field. When I started, it was simulations took a really long time, and you had no idea what parameters you really needed to test in order to make agreement with the data. Now we're in a regime where we can actually simulate these networks in a very fast and efficient way on standard workstation computers, and also find parameters in an automated way that allows us to make predictions about experimental data. So in that way, I think that there's an interesting opportunity for spiking neural networks over the next few years, and also specifically through the hardware that's being made available by Center for Analytics.
Which in this case is quite large memory. So maybe not accessible to everybody, but it is standard sort of CPU type hardware. We're not expecting, you're not leveraging large GPUs.
Definitely. Yeah. So you need a large amount of memory, but as that's becoming cheaper and cheaper, it's becoming more and more accessible.
Yeah, that's fantastic. Great. Well, thank you everybody for joining us today. We're near the end of the hour, and thank you especially to Lyle, Lawrence, and Mohammed for joining us and presenting on the very exciting work. We're very excited to continue collaborating with you and working with you on supporting this great initiative and the potential impact that it could have in ultimately potentially controlling seizures, as Lyle and Mohammed described earlier. So thanks again, everyone. We have the recording of the session, and we'll be sharing recording with everybody who's registered online via a follow-up email. So thanks again and have a great day.